So, after listening to the previous content in this lecture, this is a good time to pause and think about things and check your learning a little bit. Here are some questions that you can use to kind of calibrate your notes and check your understanding. You should be able to list the differences between RNA and DNA at the nucleotide level and also at the entire macromolecule level. You should be able to label the 5' and 3' ends of a strand of RNA. You should know the purpose of messenger RNA and aspects of its structure, like is it single-stranded or double-stranded? Single-stranded. Does it base pair internally? And the answer to that is no, and so on. You should know what the purpose of transfer RNA is, and you should be able to describe and recognize aspects of the structure of transfer RNA. For example, you should know that internal base pairing is important, and that on a transfer RNA molecule, you have an end that will hold amino acids, a specific amino acid for each type of specific tRNA, and that on the other end, there's a 3-nucleotide sequence called the anticodon, which is what's going to come in and dictate whether that tRNA actually sticks in a ribosome and whether the tRNA then drops off its amino acid. We'll look at that more when we learn more about translation. So let's move on now to rRNA. R stands for ribosomal RNA. So shown here are two drawings or models of a ribosome. They kind of look like hamburger structures. Here's a ribosome, and here's a different way of drawing a ribosome. Ribosomes make peptides, so they catalyze the synthesis or the formation of peptide bonds between amino acids. Here's an amino acid. Each little green blob is an amino acid. In between each of them is a peptide bond, and the ribosome is making that happen. Okay? Here are the transfer RNA molecules that we just discussed, shown in blue, green, and red. That's their anticodon region. Just talked about that. Okay? And each of them was carrying in these amino acids a moment before. This is messenger RNA. So where's the ribosomal RNA? Well, the ribosomal RNA in this drawing is just part of this golden mass. Okay? It's shown here, again in gold, as these little nucleic acids. That's our RNA. Okay? It's part of the structure and function of the ribosome. So you build ribosomes out of rRNA. And in fact, it is the rRNA that makes the peptide bonds. Okay? The rRNA does all kinds of things here. It dictates the structure of the ribosome. It holds it together. It's what actually catalyzes the formation of these peptide bonds. And it also helps to position messenger RNA on the ribosome or in the ribosome so that translation can occur. Thinking a little bit more about ribosomes, what I should have mentioned when we were looking at this is that this large part of the hamburger ribosome structure is the large subunit. This is the small subunit, and that ribosomes are made really similarly in all living creatures. There are two basic patterns, the type of ribosomes that happen in prokaryotic cells and the type of ribosomes that happen in eukaryotic cells. Here's the hamburger-like intact ribosome here and here. Eukaryotic ribosomes are a little larger than prokaryotic ribosomes. All of them are made of a mixture of protein and RNA, rRNA and R protein. So this is a molecule of ribosomal RNA. This is a molecule of ribosomal RNA. Combined, they contribute to the large subunit of this ribosomal structure. Here's another molecule of ribosomal RNA. It's found in the small subunit. And then we've got a mixture of ribosomal proteins not named here. In the eukaryotes, we've got the 28, the 5.8, and the 5, 3 different molecules of rRNA, all contributing to the large subunit. And again, another 1 molecule of rRNA contributing to the small subunit. So they're part of the structure and function of ribosomes. They have very complex 3-dimensional structure, something that was only solved in the last decade or so. Our understanding really grew about ribosomes and their structure in the last decade or so. This is an article that talks a little bit about that. So this is Saccharomyces cerevisiae, which is a eukaryote, and it's the large subunit and the small subunit. And so this is how the molecules of rRNA that I mentioned here, another way of drawing them or showing how they're organized. And you'll notice they're kind of like all fused together, right? And then this is the small subunit. There's the 18S and all its different regions that perform different functions. That's found down here in the small subunit. Okay, so a thing to remember is that base pairing can happen within a single strand of RNA and between different strands of RNA. And I want to mention this now, and I'm going to mention it again, and this should say not transcription, but translation. Let's get this right. Okay, so here's the hamburger ribosome-like structure shown here, and this is time from left to right. Okay, so in order for a ribosome to translate a molecule of messenger RNA, which is what this gray line is here, okay, the ribosome has to assemble on top of the messenger RNA, and then it has to move down the messenger RNA. And as it's moving, it has to build the peptide, and that's what the blue stringy stuff is coming out the top. When the ribosome gets to the end of the messenger RNA molecule, or close to it, more accurately to a stop codon, the ribosome comes apart and off goes the peptide. And so what I want to mention right now is that this simple drawing is relying on the activity of the rRNA within the ribosomal subunits. So I'm going to point out two ways it does that. In this small subunit of the ribosome is a molecule of rRNA, and it is what actually attaches to the messenger RNA at the correct position, a ribosome binding site, getting everything centered and appropriately placed so that translation can occur correctly. Base pairing is happening there. The rRNA in this little pale beige blob is base pairing with the messenger RNA in this gray messenger RNA line. A's are base pairing with U's, C's are base pairing with G's, and vice versa. Once that happens, okay, once the small subunit attaches to the messenger RNA through base pairing, then the large subunit attaches, and then synthesis of the polypeptide occurs. And the synthesis relies on the ribosomal RNA in the large subunit, which is catalyzing the formation of peptide bonds that are making or building this polypeptide. This is an example of catalytic RNA, when RNA can act as an enzyme. Okay, so the messenger RNA is playing a role in translation because the messenger RNA is telling the ribosome which amino acid to place here. So messenger RNA is informational. Okay, the transfer RNA shown here, 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 and here, it's playing a structural role, and it's a carrier. So it is working to carry the correct amino acid into the ribosome. What the tRNA that gets in is controlled by base pairing between the anticodon region and the codon on the messenger RNA. Anticodon on tRNA, codon on messenger RNA. Meanwhile, within this blob that is the ribosome, we've got ribosomal RNA, which is actually catalyzing the formation of the peptide bonds between this growing peptide, and also helping to do other work, like it contributed to the initiation of translation, as I mentioned a moment ago. All right, so the three major categories of RNA in cells are messenger RNA, transfer RNA, and rRNA, and we've just talked about their structure and function. There's an additional category of RNA that has been discovered and studied in the last 10 to 15 years, and it's a category called interfering RNA. Within that category, we have microRNA and small interfering RNA, miRNA and siRNA. Basically, what these two types of RNA do is they interfere with translation. So they can actually stop or turn down translation, which allows them therefore to silence genes. So you've got the gene in the cell, you get messenger RNA, the messenger RNA goes to the ribosome, but it does not get translated because of the interference due to these molecules. This is something discovered by Fire and Mello, and it was their work resulted in a Nobel Prize actually in physiology or medicine a few years ago. So the skinning on microRNA is that within our genomes, because this is very common in eukaryotes, within our genomes we have these little genes that encode little RNAs called microRNAs. When those little RNAs are produced, they get extensively processed, they attach themselves to some protein, and then they will base pair with a complementary target messenger RNA molecule. So somewhere in our genome we've got a gene for messenger RNA, and somewhere else we have a gene for a corresponding microRNA. If we express both of them at the same time, then what will happen is you'll get base pairing between the interfering microRNA and the messenger RNA, and it will turn off translation. So we screw up the translation process so we don't get the peptide that messenger RNA molecule shown in blue there would normally produce. Small interfering RNA or siRNA works similarly. So they are also encoded in the genome, but what they do is they will target some messenger RNA in the same way as microRNA targets a specific messenger RNA sequence, and they'll actually like chop up that messenger RNA. So they won't just base pair to it and like sit there blocking translation. They'll actually chop up the messenger RNA. So they also will stop translation, and so you won't get the protein associated with this silenced or sliced messenger RNA. So again, both of these fall into a category called interfering RNA, where we've got little RNA genes, and the result of expressing them are these tiny pieces of RNA that stop the translation of other RNA called messenger RNA in a really specific manner. And I'm going to close with that. I'm just going to point out that there's this really popular dark matter is like this really popular phrase these days, and like all the different scientists use it in different ways. But people who study RNA quite a bit often call RNA the biological dark matter. They don't own that term. Microbiologists like to call all the unnamed microbes that exist in the world dark matter, but we think of RNA as dark matter because we don't know actually as much about it as we would like. So we're always learning new things about what RNA does. For example, discovering those interfering RNAs, which is really a pretty recent discovery, and also learning more what RNA does in cells and in viruses. A lot of viruses, their entire genome is made up of RNA, and many of them haven't been even discovered yet. We've just seen evidence that they exist based on sequences recovered from the environment, and we're still discovering new ones all the time. Another category of dark matter is new viruses that we haven't yet discovered. So with that, I'm going to close this lecture on RNA structure and function.